developmental neurobiology, so neural ectoderm formation. So let's just get started. So today I want to talk. I want to discuss some points that I need to finish up in the discussion of gastrulation. As you know, gastrulation is all about laying down the germ layers, bringing everything, and bringing the mesoderm and endoderm inside the embryo, and then setting in place the conditions for differentiation. This is a flowchart for what each tissue gives rise to. And today we're going to talk towards the end about the mesoderm part. As I mentioned, Sonia and Bhavna will talk about ectoderm and neural tube differentiation. And I think Arjun talks a little bit about endoderm differentiation. Okay, so this is gastrulation happening right now. And there's and it looks from the discussion on Wednesday, it looks like a relatively easy process. All we have to think about are cells moving in, perhaps EMT happening, and then things like that. But there's actually quite a lot to unpack here. Let's just go through this one more time. The cells, the mesodermal and endodermal cells, are moving in through the streak, and this is the streak here. They're moving in through the anterior streak as well. But what I want to talk about today is kind of these different phases of gastrulation. And so you can start seeing at this point that the streak and the node that's at the end of the streak starts moving back. And so these are different properties of the streak and the node that happen at different times. And so this is best shown, I think, in the next slide. What we first want to understand is how the streak forms. And so, as you all know, the streak is the point at which the point of ingress for the mesoderm and endoderm. It forms somehow, and the next image just shows what these properties are. And so this is the chick, and this is in a newly laid chicken egg. And what you're looking at here are the movements that make the streak. And so you can see that the epiblast cells reorganize on different sides and swoop down to form a streak here. Let's see this again. And so these movements that the epiblast cells are making are called Polonaise movements. Polonaise is a dance, and it's danced by dancers with long skirts that swirl when they turn around. And so this swirling is happening here, and it's forming the streak in the center here. And this reorganization of the cells is happening by anisotropic intercalation, driven by planar cell polarity genes. And so this is kind of shown here. You can see this, the cells of the epiblast reorganizing, so that you have a kind of a streak primordia that's quite short and fat. And as the cells intercalate, this streak becomes elongated and quite nice. This video from Fija's group is of cells that have been infected or transfected with the GFP, so that some of the cells are fluorescent. And then what they've done is to track the cell movements. And you can see this Polonaise very beautifully here. And so these are comets of where the cell trajectories. And you can see very nicely this kind of movement that's happening as the streak in the chick is being set up. This is probably the same for the mouse as well. It hasn't been shown in such great detail, but the mouse has other nuances that we won't cover in this talk, so in such detail. So once a streak is set up, it elongates, and it elongates to a particular length. So the streak will form a very nice primitive streak, and the most anterior part of the streak is called Henson's node. Henson's node is equivalent to is slightly equivalent to the blastopore lip in Xenopus, and I'll show you why that's the case in the next slide. We'll talk about the movements that are happening through different parts of the streak in the next few slides as well. So Henson's node acts as an organizer in the chick. It's believed. So Waddington did these very nice experiments in 1933, where he took the node from a duck embryo and transplanted it in a chick embryo. And what he found was that he could induce a second neural tube. So he could induce another axis within the embryo. So here you're not seeing a duplicated axis, you're seeing a proper second axis. Claudio Stern repeated these experiments using more modern labeling techniques, and found very nicely that the second axis contained both forebrain regions in red here, and in red in the induced structure, as well as more caudal neural tube structures labeled in blue here. And so this there's a slight nuance to this as well. Of course, Henson's node is giving rise to various tissues, and so what has always been unclear to me is whether it's Henson's node proper that's acting as the organizer, or whether it's the tissues that Henson's node is laying down. So this is that. Let's talk about then the steps of gastrulation. And so early gastrulation, like I said, so gastrulation happens through the primitive streak by EMT. Cells that are the epiblast cells that are lateral to the streak are constantly recruited to the streak. And so as streak cells ingress and move into the rest of the embryo, the cells that are lateral to the streak cells are told to become streak, and they are then able to go through the streak and undergo EMT. So remember the streak here, I'll play this movie in a bit, is running all the way up to here, okay? The caudal primitive streak cells, so the cells that are going through the more posterior caudal primitive streak, generate more ventral mesoderm. The ones that are going more rostrally generate more dorsal mesoderm. So anterior primitive streak, that's Henson's node, which has also been called the node streak border, generates definitive endoderm at these stages. And so at this point, while in early gastrulation, Henson's node is not actually generating any mesoderm. It's only generating endoderm. And so let's look at this. And so you can see the cells emigrating from the streak, and these the endodermal cells here. Okay, but there's something else happening as well. So at some point, these cells are producing axial mesoderm, and we'll go into the different mesoderms in the next couple of slides. And I've just told you during early gastrulation, the anterior primitive streak, or the node, is only producing definitive endoderm. So it's producing definitive endoderm here, but then at some point, the anterior primitive streak starts to regress, and you can see this regression happening right now. Okay, and as it regresses, it's starting to generate axial mesoderm. Okay, this regression of the streak as it's being brought down, I think of as kind of more mid to late gastrulation. Let's think about that here. So let's play this movie, and then I'll talk you through it. So what's happening here is the authors of this preprint have put in 2 spots of dye. So in the previous image, they had put this red dye. This is a dye called dye I, which is lipophilic, in these 2 spots within the streak at a particular stage that represents early gastrulation. Here, they're putting it slightly later. They're putting a dye O, which fluoresces green, and a dye I spot next to each other. And what they're asking for, what they're trying to see, is whether these lateral cells ever contribute to the mesoderm. So whether these lateral cells will go towards the streak and then fan out as they go through and then become mesoderm. And what you can see is that the more caudal spots here and here they become recruited into the streak and then become mesoderm. Like this, you can see this happening here. Whereas this more anterior, more rostral spot here, this one, does not go through the streak. Okay? And this becomes more neural tissue, or it becomes neural. Okay? And so let's go through what's going on here. So the lateral epiblast cells, so during mid to late gastrulation, these lateral epiblast cells are no longer recruited to the Hensen's node, or the neural streak border. And when these lateral epiblast cells fail to be recruited, the Hensen's node starts to retract. As it retracts, the node starts generating axial mesoderm, and it also generates the ventral neural tube. The epiblast lateral to this now streak. So the streak can be also thought of as a mesodermal stem zone. So it's generating mesoderm constantly. And so the epiblast lateral to this region is now specified as neural. And so lateral epiblast now can form kind of non-exclusively, so it's still a little bit noisy. It forms caudal neural tissue. So what I'm saying here is that the epiblast, let's stop this a little bit. This epiblast here, around the around the neural stem cell border, the node, is now specified as neural. And so as retraction happens, this lateral epiblast is almost exclusively neural, and will form, will start forming the neural tube. Okay. So this is mid to late gastrulation. We still have late gastrulation happening as well. And so gastrulation continues to generate the axis. And so the embryo needs to elongate over long periods of time. And there is still a pocket of gastrulation going on even at later stages. And this is in the tail bud. And the tail, and so this review here, which I'll put up at the end of this lecture, talks about axis extension happening through the tail bud. We have a continuation of the neural streak border that is retained as the embryo elongates. The lateral epiblast here, next to the remnants of the streak, it constantly puts down neural ectoderm as well. And so these this kind of interaction is required for the extension of the embryo. I don't want to go into this in too much detail. We have we have more to talk about. But if anyone is interested in this, if you have a question about what I've talked about so far, please ask it. Otherwise, I will put up details on this in the Moodle page for this lecture. Okay. Cool. So here, here let's look at the extension closer detail. And so you can start seeing the streak beginning to retract like this, and also the first vestige of mesoderm starting to differentiate. And this we all know about are the somites here. Okay, you can see somites condensing here as more and more mesoderm is being generated. So let's talk more about the mesoderm. But first, I should say that there is, so the reason why we've talked so much about chick is that the ability of the chick embryo to be imaged. And so what we're trying to talk about is amnio embryos. The chick is flat. It can be cultured ex vivo very easily. And so many of these images are of the chick. The mouse, of course, is much more difficult. It develops inside the mother. It needs particular types, and its geometry, this cup shape, makes it really quite difficult to image. A couple of years ago, Philip Keller in Janelia did set up very beautiful imaging techniques for the mouse embryo. And so we'll have a look at this right now. And so this is the mouse. You can see that it's a cup shape here. And so this is a lateral view, a frontal view, and a lateral view. And this is a ventral view. This is the mouse's equivalent of Henson's node, which is here in the mouse just called the node. And you can see this beautiful development happening in the mouse. Again, there's some reorganization of the epiblast going on ventrally. The streak is forming. It's just up here, and here the what will become the head of the mouse is there. Okay. Okay. So all of these movements are laying down, are bringing in the mesoderm and the endoderm into the right place. So for the rest of the lecture, I'm going to talk about the mesoderm, and so this is going to be a run through all of these different parts of the mesoderm here. Okay, so before I go into this, if you have any questions about gastrulation per se, can you ask them? Okay. Okay, good. So we have essentially 4 different zones of mesoderm that are laid down. There's axial mesoderm, there's paraxial mesoderm, intermediate mesoderm, and lateral mesoderm. The distinction between intermediate and lateral mesoderm is not so great. Many people incorporate intermediate and lateral mesoderm together, but we'll separate it because they give rise to different structures. What we're talking about is a mesoderm that's progressively more and more ventral. So axial is the most dorsal mesoderm. Paraxial is more ventral, even though we're going laterally. When the embryo finishes its morphogenesis, all of these mesoderms become ventral. Intermediate is the next more lateral, more ventral, and then lateral plate mesoderm, lateral mesoderm is the most ventral. Okay. So let's talk about the regional specification of mesoderm. And so this can be seen here in the streak. And so already within the streak, there's some specification of these different zones of mesoderm. You can see here now the sorry. You can see here the most rostral part of the streak giving rise to axial mesoderm. A little bit lower down, this mesoderm is starting to form. Let's go back. So you can see this spot here, generating this mesoderm here. And this is more paraxial mesoderm. And then this caudalmost spot here, generating more ventral, more lateral mesoderm. And this will probably form in a lateral plate mesoderm. So already within the streak, there's a pattern to the mesoderm that's going to be laid down. And so there's I will direct you to a paper that I'll put up in Moodle, which talks about the patterning within the streak. And this paper is from Olivier Porquis' group, which talks about nested expression of Hox genes within the streak. And so regional specification of mesoderm. So how the mesoderm is being laid down is probably already happening within the streak. The most anterior mesoderm, the most anterior streak will give rise to axial mesoderm once the streak starts to regress. An axial mesoderm forms 2 structures called the precaudal plate and the notochord. Here is a human embryo. The precaudal plate is a mesodermal structure, although it's quite intermeshed with endoderm as well, and so its exact And so in both in human, mouse, and chick, precaudal plate is generally termed mesendoderm because it's difficult to separate both endoderm and mesoderm from the precaudal plate, and it's because of this what we're seeing here. So when axial mesoderm is generated, it's quite contiguous with the endoderm. The notochord itself will segregate very nicely from the endoderm, whereas the precaudal plate retains this kind of structure, or this kind of structure here. Of course, notochord and precaudal plate to some extent does have a role in later development, but at these stages, it forms essentially patterning centers. Precaudal mesoderm or mesendoderm can induce anterior structures, and so this is the nuance of the Henson's node transplant experiments that I wanted to get across. And so here, what Kessel's group have done is to transplant precaudal plate into ectopic locations within the embryo, within the chick embryo. And what they're finding is that they can now induce a second forebrain within the chick. And so this transplant in ABC is a single transplant, and it causes now an ectopic forebrain structure, double transplants or transplants of the precaudal plate and the slightly more posterior notochordal plate here will cause, so only the precaudal plate will induce anterior structures, the slightly more caudal structures will not, and so on. Okay. So precaudal plate seems to induce anterior structures, and it probably is used even after emigration to somehow modify the response of the neural tube to forebrain inducing, forebrain induction. And hopefully Bhavna and Sonia will talk about this next week. The notochord patterns the neural tube. And so it's known that the notochord emits particular signaling factors. It emits sonic hedgehog, which will induce ventral structures within the neural tube. And these lead to the generation of motor neurons within the neural tube. So the notochord is playing an important role in neural tube formation. It's also playing an important role in the differentiation of somites as well. So the notochord is patterning somites in a particular way to form particular substructures of somites, and we'll go into this in subsequent slides. But it's imparting pattern towards the more paraxial structures as well. So notochord is important for patterning within the embryo. Before I go on, the notochord does form something in adults. Does anyone know what the notochord will give rise to?
From the vertebral column.
Yeah, almost. So it's not quite the vertebral column. The vertebral column actually forms from somites. And we'll talk about that in the next couple of slides, Shubha. Okay. The vertebral column actually forms from sclerotome. But I'll show you what the notochord gives rise to as well. So somites are, we've looked at somites and we've discussed how they form these periodic structures a few times now. And so somites are these blocks of mesoderm. They're paraxial, they're classic paraxial mesodermal structures. And they form the vertebrae, and they form the muscles of the back. They form almost all the muscles of the of the of the animal. So paraxial mesoderm gives rise to the musculoscheletogenic system. It forms from somites very beautifully. This very nice review here talks about muscle formation. I don't have time to go into the details of muscle development. Muscles have been classically looked at, and so a lot is known about how muscles form, including kind of the genetic pathways that lead to muscle differentiation. If anyone is more interested in muscles, we can have a discussion about it at a later date, or we can ask Dhananjay from Vijay's lab to come and talk about it. But so somites give rise to the musculoscheletogenic system, and they are forming from paraxial mesoderm. When the somites form, they're subjected to, so they form as kind of condensed blocks of mesoderm, and they're subjected to information coming from both the surface ectoderm and the notochord, which gives rise to asymmetries within the somite. And so the somite will start forming kind of substructures. So what we will start seeing is this structure here that's labeled DM, which is the dermamyotome, and SCL, which is the sclerotome. So dermamyotome will give rise to essentially muscles, so give rise to myotome. And it also gives rise to some extent to some of the dermis as well. Sclerotome will condense to form the vertebral column, as well as some other structures as well. There are, even within the dermamyotome, there's a dorsal dermamyotomal lip, a ventral lip as well. And these form emigration points, for muscle precursors to start migrating across into different parts of the animal as well. And so the muscles that form around the gut, for example, or in the limbs are all generated in these structure in the dermamyotome, and they'll migrate and populate different regions of the embryo. So the sclerotome will give rise to the vertebral column, and it does it like this. So the sclerotome will migrate around the neural tube and will start to encase the notochord. And so the notochord becomes then encased in the sclerotome, and it will also start forming these processes that wrap around the neural tube as well. And so the vertebral column is more or less segmented, and that these segments come, of course, from the somites. The notochord ends up in the middle of the vertebral column and starts to contribute to the intervertebral discs. And so the last vestige of the notochord in us is in the center of the nucleus pulposa, which is part of our intervertebral discs. Okay. So this is vertebrae from paraxial mesoderm. Intermediate mesoderm gives rise essentially to the urogenital system, so kidney and gonads. The embryonic kidney, and so we'll talk about the kidney more than the gonads. I'm thinking to ask someone to talk about gonadal differentiation towards the end of this lecture, but I'll see if I can catch him and ask him to give a lecture. So I'll talk about kidney at the moment. So the embryonic kidney actually derives from 3 primordia, all derived from intermediate mesoderm. So these are called the pronephros, mesonephros, and metanephros. And so these are at different rostral caudal extents. So the pronephros is fairly rostral, mesonephros a little bit more caudally, and then the metanephros more caudal than that. The adult kidney actually forms from the metanephros, so the pro and mesonephros degenerate during embryonic maturation. The kidney is a composite of the so it forms from metanephric mesenchyme. So the kidney proper forms from metanephric mesenchyme and an outbudding of the Wolffian duct, which is called the uteric bud. And so these 2 structures develop in concert to form the kidney. And this is kind of seems to be a innovation, not so much of an innovation, but it seems to be amniote specific, because in teleost fish and amphibians, it's actually the mesonephros that functions as a kidney in those animals. Again, the development of the kidney is fairly well worked out. And so the kind of the transcription factor pathways that are in play are known. Nishi Nakamura's group from Kumamoto in Japan have worked out very beautifully how to make kidney structures from stem cells as well. And so they're able to manipulate the developmental pathway ex vivo to understand how this structure starts forming. And so this is very useful. I recommend you to have a look at Nishi Nakamura's work. So his work, and also the work of Andy McMahon, who's been looking at the development of the kidney in the mouse to understand then what we have to think about when the kidney develops. And so, of course, these are clinically important as well. There are many congenital defects that affect kidney function, and a lot of them can be traced to defects in, for example, the metanephric mesenchyme being able to condense. Okay, so this is intermediate mesoderm. The next is lateral plate mesoderm. And so lateral plate mesoderm is split into, so in most animals, you have this kind of split in the lateral plate mesoderm around the coelom of the animal. You have what's called somatic lateral plate mesoderm and splanchnic lateral plate mesoderm. So somatic lateral plate mesoderm gives rise to some of the extraembryonic mesoderm. And so these will form blood vessels that carry food, either in form of the placenta or that wrap around the chick egg, and they'll form blood vessels that will feed the embryo. It contributes to the dermis as well, to the limb skeleton, and to the pelvis. And so the somatic lateral plate mesoderm is giving rise to all of these. It also, to some extent, forms the body lining. So it forms the pleura and peritoneum and pericardium. Splanchnic mesoderm forms essentially the visceral lining of the organs. You'll get hemangioblasts from splanchnic mesoderm. So hemangioblasts are, of course, not just hematopoietic precursors, but also things like endothelial cells as well. So the vasculature system comes from splanchic mesoderm. The heart forms from splanchic mesoderm. Mesenteries, the lymphatic system, the lining of the gut and the lining of the lungs are all derivatives of splanchic mesoderm. The kind of individual differentiation of these structures, I'm not going to go into. The heart, Soumyashri Das will talk about in about 7 lectures time, six or seven lectures time, and so she'll talk in more detail about the heart. But I just wanted to make you aware that there are these that there is somatic and splanchnic mesoderm. They both are derivative. They're both lateral plate mesoderms, and they're forming different kinds of structures here. So what I will finish with is a small discussion about that there is another difference. And this is the difference between head and trunk mesoderm. We know that the paraxial mesoderm of the trunk is segmented, and so this is so these form somites. And what you're looking at here is a chick embryo where the ectoderm has been removed, and so you're looking at kind of the mesoderm kind of exposed for us to see. Here, and so this is this brings me to a kind of interesting controversy that happened a long time ago that I think is now resolved. Whereas trunk mesoderm is segmented, cranial mesoderm, so this is the mesoderm that's in the head, is unsegmented. There's an ongoing, even now there's an ongoing controversy, which there's less and less subscribers to, which suggests that there is some cryptic segmentation in head mesoderm, and these are called somitomeres. And here, this is from Stephen Meyer, who's drawing out these segments in the cranial mesoderm. Most people accept that there's no such thing as somitomeres in the cranial paraxial mesoderm, and that the cranial paraxial mesoderm is unsegmented. So already now, if we think about the lack of segmentation, or even if it's cryptic segmentation, there's a huge difference between what's happening in the head and what's happening in the trunk. The lateral plate mesoderm as well is it has acute differences in the head and the trunk. And so whereas the posterior lateral plate mesoderm gives rise to things like blood and blood vessels, only the anterior lateral plate mesoderm is able to give rise to the heart. And so so these kind of differences arise at some point in development, and it is known that you can move cells from within the epiblast to a different location around the streak, and they'll contribute very nicely to heart mesoderm. And so this is happening. It seems to be happening after emigration of the mesoderm, while the embryo is being set up. And so the mesoderm is being patterned after it's being generated as well. And so it's being patterned in an anterior-posterior fashion. What those mechanisms are is still a little bit unclear. This is a review from Ramkumar, who was in STEM and now in ISA Tirupati. You can have a look at this. So what the review says very nicely, it talks about these differences in head and trunk mesoderm. The head is unsegmented will give rise to different structures in the pharynx and in the heart. The trunk will give rise to different structures in the somites, the kidneys as well, and the blood vessels of the body. And he suggests that there are, of course, signaling factors that are different between the head and the trunk. The head is full of antagonists for various signaling factors, for nodal, for example, and for Wnt, are expressed very highly in the head, whereas the trunk seems to be a region where FGFs are expressed and Wnt genes are expressed. And so already we can see some asymmetry happening between the head and the trunk, and this asymmetry is reflected in what head mesoderm and trunk mesoderm will give rise to. And then these then go on their own differentiation programs to form the rest of the body. Okay, I think that's my last slide. Yep. So that's my final slide. If there are any questions, please ask them now. Cool. You can still hear me, yeah?
Yes, I had a question.
Yes.
Okay, thanks, Nivedita.
Yeah, so you were talking about the somatomeres and how there's a debate regarding the same. So could you, do you know what led the authors to believe that these are segmented? Like, what is the basis of this segmentation?
So, sorry, I'll share the slide again. So they believe that there are. They've drawn around what they think are segments in the cranial praxial mesoderm, and so they're seeing bulges in this cranial praxial mesoderm. And then they're saying that these are kind of bona fide segments, and so like somites here, these somatomeres may not mix with other somatomeres. What many people have done since is to try and label individual cells within the somatomeres and show that these cells don't care where they are. They'll mix very nicely up and down the embryo. It's possible that what these bulges are reflecting are bulges that happen in the neural tube. And so the neural tube, of course, is around the paraxial mesoderm, and the neural tube does have bulges. It forms what's called rhombomeres, which are and these may be pushing the somatomeres into these structures. Okay.
Okay, thanks.
Cool. Okay. So if there's no other questions, I'll let you go on with your day. As I said, I will put up this lecture onto Moodle. I'll give you some reading to do as well, and you should all start thinking about your end of course essay as well, and I'll put up the instructions for that this weekend. The deadline for that assignment will be sometime in January, so you've got a long lead time, but don't leave it until the end of January. Okay. Cool. Okay, so thank you, everyone. Have a lovely weekend.
Thank you, Raj.
Thank you, Raj.
Thank you, Raj. You too.
Okay.
Bye-bye.
Bye-bye.